Hello viewers, welcome to my YouTube channel. Today I am going to start a very relevant topic which is commonly used in the biology. So the topic for today is cell transport. Better we can say membrane transport. So how the things move in or out of the cell through the cell membrane, that is actually the cell transport or membrane transport. So let's start about its types. So this is the types of cell transport. Cell transport is divided into three parts. Three types are there. First, the passive transport, active transport, and vesicular transport. Passive transport is actually which is where the molecules throughout the membrane they travels down the concentration gradient. Here no energy is required, and also that is the main difference between the active and passive transport. So I am coming in details. First look over the chart. Passive transport, it is divided into four: simple diffusion, facilitated diffusion, osmosis, filtration. Active transport, which is divided into two: primary and secondary. Secondary, which is also divided into uniport, symport and antiport, and vesicular transport, that is divided into two parts: endocytosis, exocytosis. And then further, the endocytosis divided into three: pinocytosis, phagocytosis and receptor mediated endocytosis. So these are all about the different types of cell transport. Now I am coming in details in different types of cell transport. First I am describing about the passive transport. So this is the first type of passive transport, simple diffusion. So simple diffusion is a type of transport where the molecules comes from the outer membrane to the inner membrane by the concentration gradient down to the concentration gradient. That means from higher concentration to the lower concentration. And no energy is required here, no carrier protein also required here. So this is the simple diffusion. Now the simple diffusion is of two types. Here it is. Simple diffusion is of two types. This is the one where the molecules are travels along the cell membrane, and some molecules travels through the channels. Generally urea, gaseous like oxygen, these are travels through the lipid bilayer, against the down to the concentration gradient. That means from the higher concentration to the lower concentration. So the molecules are traveling from the higher concentration to the lower concentration across the lipid bilayer. This is one type of simple diffusion. And the another one is ions which are traveling from higher concentration to lower concentration through their specific channels. Now the question is that why ions are not traveling through the lipid bilayer? This is because these ions have their own charge, and lipid bilayers are also have their charge. So they are actually because of their charges, they are not able to come across against the by the lipid bilayer. That's why they have their separate channel, and ions are moved through these specific channels. So this is the simple diffusion where molecules are travels from higher concentration to lower concentration through the lipid bilayer, through the membrane, and these are of two types. One that is gaseous urea, that kind of molecules are crossing the membrane down the concentration gradient by the lipid bilayer, and another one, some molecules are traveling through the specific channels. So that is simple diffusion. Now Now one more thing I have to mention here, that is these ion channels are of two types. One, voltage gated, and another one, ligand gated. Voltage gated and ligand gated. Voltage gated and ligand gated. These are two types of ion channels. Voltage gated ion channels are open. Actually opening and closing of the channels depends on the charge. Developed across the membrane. And ligand gated ion channels, here the channels opening and closing of the channels depends on the chemical. Okay, here the voltage gated sodium channels are there, is an example, and acetylcholine, glutamate, these are example of ligand gated channels. So the simple diffusion is of two types. This is one, and this ion channels is the second. These ion channels are also divided into two types, that is voltage gated ion channels and ligand gated ion channels. These are opens by the charge developed across the membrane, and these are open depends on the chemical. The opening and closing of these channels are opens depends on the chemicals. The second type is facilitated diffusion. Facilitated diffusion, here no energy is required, but protein carriers required here. It actually follows the ping pong mechanism. That means this is the carrier protein. They receives the ping pong mechanism is that these proteins are receiving the molecules. After when the molecules get attached with that protein, the protein then carrier protein, then conformational changes in the carrier protein, conformational changes occur, and after that the molecules get entered into the inner side of the cell. This is the outer side of the cell membrane, and this is the inner side of the cell membrane. In all the passive transport, the molecules are moving from their higher concentration to the lower concentration. Here is one type of passive transport, that is facilitated diffusion. This is here the thing is also same, higher concentration gradient at the outside of the cell membrane, and these molecules are moving through the carrier proteins. These carrier proteins are receiving the molecules, and then it passes this molecule into the inner side of the cell membrane. So here the traveling of the molecules are traveling through the carrier proteins. Okay, these are the protein channels. So that is a facilitated diffusion. These are actually of two types, uniport and antiport. Uniport, when the only single molecules can move. Only one single molecule can move through the protein channels. And antiport is when the one molecules get entered into the cell, then another molecule from the inner side goes to opposite direction. That is antiport. So these are also comes under the active transport, but they are energy required. But here in the passive transport, uniport, symport are actually the type of transport where no energy is required. So these are the facilitated diffusion. Uniport example here is the absorption of sucrose from the intestine, transport of glucose in the skeletal muscle. So these are some examples. And the chloride bicarbonate exchanger, which is present at the gastrointestinal portion in stomach. It is also responsible for HCl secretion. Okay, and chloride shift in RBC. These are all actually the example of facilitated diffusion. Now come to osmosis. In the osmosis, the solvent molecules are moved from their higher concentration to the lower concentration. Look, one thing we have just studied that in simple diffusion and in facilitated diffusion, the solutes are coming across the membrane. Okay, Look at these pictures in simple diffusion and facilitated diffusion. In these two types, the solutes are coming across the membrane. Solutes are coming across the membrane. Okay. But in osmosis, the solvent molecules are coming from the higher concentration to the lower concentration. Okay, look at the picture here. These solvent molecules are higher. The concentration of the solvent molecules are higher in this part, and here the solute molecules are higher, but the solvent molecules at lower concentration of solvent is here and higher concentration of solvent is here. Okay, solvent, that means water molecules, and sugar is, you can consider as a solute. Now after osmosis, look at that picture. This is the semi-permeable membrane. This is semi-permeable membrane. Semi-permeable membrane. Then after osmosis, all the solvent molecules goes to this part. Okay, through the semi-permeable membrane. So here the solvents move to the lower from higher concentration to the lower concentration. That is osmosis through the semi-permeable membrane. Now it is filtration. Filtration. It is depends on the mechanical pressure gradient, which force the fluid to go from higher pressure area to lower pressure area. Okay. It is actually important in the transport of nutrients and water to the tissues, capillaries from the higher pressure region to the lower pressure region. So the pressure terms is very much related to the filtration. Look at the picture. These are the molecules. These molecules under pressure, there is a much more molecules, so it's a higher pressure region, and here less molecules are there, lower pressure region. So from the outer side, by the pressure gradient, molecules moves through these parts, come to inner side. So from the higher pressure region, the molecules come to the lower pressure region. That means the molecules are filtered out. Okay, so that is filtration. That actually some activities like cardiac activity, gravity, this triggers the filtration. So here we discussed about these four types of passive transport: simple diffusion, facilitated diffusion, unipode, antipode, osmosis, and then filtration. So these are four types of passive transport. Now I am coming to active transport. In active transport, molecules are moved from lower concentration to the higher concentration. Okay, and energy required here, and also it's a carrier mediated. That means carrier also required. And energy requirement is actually of two types, direct and indirect utilization of energy. So the active transport is mainly based on the energy. Without energy, active transport couldn't be happen. So look at the picture. This is the primary active transport. There are two types of active transport, primary and secondary. So first I am explaining the primary active transport. In primary active transport, energy, here this is a channel. We call it as a sodium potassium ATPase. This channel we can say it as a sodium potassium ATPase is a protein. Okay, that this pumps out the sodium to outside. Pumps out the sodium to outside, and at the same time it pumps in the potassium at the inner side. How it's possible? It's possible by the hydrolysis of the ATP. At that time, ATP hydrolyzes to form the ADP and PI, and here the energy is liberated. Using that energy, that pump throwing the sodium to the outer side, and also it get enter the potassium get enter through the channel at the inner side. Okay. There are other proteins are there like calcium ATPase, sodium hydrogen ATPase. So these are some proteins that facilitate that primary active transport. So here the utilization of energy is direct utilization of energy from the ATP. Now the second type of active transport, that is the secondary active transport. Here it is of two types. Here the utilization of energy is indirect. Okay, so these are two types, symport and antiport. In case of symport, look at the picture. These are the transportable ions. Two types of ion. Okay, two types of ions are getting entered, and they actually traveling through the protein, through the channel at the same time. Two types of ions. Okay, this is the symport. That symport is also called co-transport. Symport is also known as co-transport. Look at the picture. Sodium ions is getting entered into the cell across the cell membrane. Along with glucose is also getting entered with the sodium ion. Okay, so sodium and glucose, these are called the symporter. Okay, they are together entered into the cell. So this kind of transport is called the symport or co-transport. Here, along with the sodium ion, glucose move inside the cell, and sodium, here the sodium is pumped out by the channel utilizing energy. Here the sodium get entered into the cell, and then what happens? This sodium is pumped out by the pump. Here is a pump. That pump throwing the sodium outside of the membrane. How they get energy? They get energy by the ATP. Here the ATP hydrolyzes to form the energy, and it forms the ADP and PI, and the energy produced by the hydrolysis of ATP utilized here. And by this energy, whenever the sodium pumped out, then in the next cycle the sodium comes in the cell. So it is an indirect utilization of energy. Okay, so this is the co-transport or symport. Now this is antiport, the another secondary active transport. In antiport, when the one molecule moves inside the cell, another molecule moves opposite direction. One molecule moves inside the cell and another molecule moves in opposite direction. Okay, and this is also called the counter transport. Okay, sodium hydrogen and calcium hydrogen, these are the channels. This type of transport is actually balance the acid base. It is actually used in acid base maintenance, balance of the acid and base. So here the sodium is getting entered into the cell, and that same thing is happening. There are channels that channels through the sodium outside of the cell. Here the energy is used by the ATP, and that energy also to fill up that space. Here the sodium is, for the next cycle, sodium get entered into the cell. So here the energy utilization is indirect, and as it's a antiport, so here the potassium ion getting entered into the cell. Okay, and potassium or hydrogen ion getting entered into the cell, and that hydrogen ion also thrown out by the cell in this way. So this is the antiport, where one molecule is getting entered and the another molecule goes out to the opposite direction. Now in this chart we have completed active transport, primary, secondary, symport, antiport. Now I am coming in vesicular transport. Vesicular transport is what? Where the molecules are transported getting entered into the vesicle. Okay, so vesicular transport is of two types, endocytosis and exocytosis. So here is the first type, that is endocytosis. In the endocytosis, the endocytosis is of two types. Okay, phagocytosis and pinocytosis. Phagocytosis is called the cell eating. It is also called cell eating. And pinocytosis is called cell drinking. Cell drinking. Because in phagocytosis, the particles are large macromolecules, and in pinocytosis, it's a fluid, liquid substance get entered into the cell through the membrane. So that is the pinocytosis, and it is phagocytosis. In phagocytosis, the particles as they are large, they often called the macromolecules or macrophage. That kind of things actually get phagocytic by the cell. So what happened? In phagocytosis, the cell membrane form a projections. That projections is called the pseudopodia, and the macromolecules or macrophages, they get entered into the pseudopodia. Okay, and then they form a vesicular structure that is also called the phagosome or food vacuole. In that way, that particle is engulfed by the cell. That is phagocytosis. So it is a cell eating. And in case of pinocytosis, these are the extracellular fluid. Whenever they come in contact with the plasma membrane, they get entered into the cell and form a vesicle. That is, and come into the cell inside. So this is the pinocytosis. Another type of endocytosis is there, that is receptor mediated endocytosis. Okay, here on the cell membrane there are some receptors. There are some receptors that receives the particles. The macromolecules or micromolecules, they receive that particles. Okay, after receiving that particles, there are some special protein beneath the plasma membrane that called the catherine. They help the cell to engulf the proteins. So by the activity of this catherine, they form a vesicle. I am giving you a detailed diagram also, another diagram. Look at this picture. Here I give the heading pinocytosis, but the same thing is happened for the phagocytosis. This is the blue colored portion is the receptor. This is receptor. Okay, that receptor, when the proteins come in contact with the receptor, then what happened? These are the catherine protein. Okay, this is the first step where the cell membrane is there, and on the cell membrane there are receptor, and that receptors receives the protein. And for the second step, what happened? There are some proteins called actin and myosin. We all know that actin and myosins are the contractile protein. Okay, they do the same thing here. They contract the plasma membrane from this part, and that means they try to form a vesicular structure. Okay, then after forming that vesicle, what happened? They form this vesicular structure is actually formed, and they come inside the cell away from the cell membrane, and then it is called the pinocytotic vesicle. If they engulf if the protein is macrophage or macromolecules, then it would be phagocytotic vesicle. Okay, the same thing will happen for the phagocytosis. Then after forming that vesicle, these vesicles get inside the cell away from the cell membrane. So these are the mechanism of pinocytosis or phagocytosis. This is actually receptor mediated endocytosis. One thing I have to mention here, that if phagocytosis, after phagocytic vesicle, if lysosome engulf that particle, then we call them as a phagolysosome. Phagolysosome. After engulfing the particle, lysosome engulfs the vesicles, and then the lysosome goes out of the cell by exocytosis. Okay, this is from the phagolysosome. This is the last type of vesicular transport, that is exocytosis. Here what happened, this is actually found in the synaptic vesicle when the neurotransmitters get out of the vesicles. Whenever the impulse come, these vesicles generally present in the synaptic knob, and by the impulse that vesicles come slowly come to the membrane, at the end of the membrane, and then they release the enzymes or hormones they contain inside them. Then they release that substances outside of the cell. So that is the exocytosis, whenever the substances are getting outside of the cell. Okay, so this is actually a picture of synapse. The substances which are exocytosis by the cell, this is the acetylcholine, a neurotransmitter. I completed all the types of cell transport. I have discussed all different types of cell transport. I am just revise the types: passive transport, simple diffusion, facilitated diffusion, osmosis, filtration. Then I discussed active transport, primary, secondary, symport, antiport, uniport. And lastly, vesicular transport, endocytosis, exocytosis. Endocytosis is further divided into pinocytosis, phagocytosis, and receptor mediated endocytosis. So these are all about the different types of cell transport. I hope you understand very well. I can give you a clear idea about the cell transport. If you have any questions regarding this topic, then don't hesitate to ask me in my comment section. If this video helped you, then don't forget to subscribe my channel. Please like, share and subscribe. Very soon I will come with a new topic, very interesting. And thank you for watching. Till then, take care.